and decline.
So I'm hoping, thanks very much for the introduction. So I've been keeping bees, working in honeybees for about 53 years. Worked about just about every part of the aspect of the industry. So I know bees fairly well. And I'm hoping that I get to my last slide because I usually give this in about an hour and it's in 40 minutes. But, oh, well, that's all right. But next week I have to give this at the tree fruit in 20 minutes. So this is good practice for me. And nothing better to stimulate me than having an IT person say, we can't do that just before your presentation. So there's a couple of embedded videos in here that we don't have sound before. So I eliminated one and I'm gonna try and narrate over another one. But it gets to the point of stimulation. If you don't take anything away from my presentation other than this, honeybees give, and bumblebees for that matter, give diddly squat about your crop. It's about stimulation. Stimulate response. Get them turned on, and you can spend money on brood pheromones and all this other kind of stuff if you really like spending money. But if you learn how to stimulate bees, if you get them excited, if you get their yayas up, they are going to do the job for you. So anyway, with that, so first thing to know about honeybees is they are not indigenous to the United States. They come from Europe, hence we call them European honeybees, and from northern Africa. And there are some down in the southern tip, a different species of honeybees. The type of bees we typically have in the United States are Ligustica, which you see on Italy, which we call the Italian bees, and I'll refer to that more. Carnica, which is in the Alps region of Europe, and then Causica, which is in the Georgia, Publica, Georgia, Turkey area, are the primary species of bees that we have. And if you look over way on that side, a Palmelia, it's a brand new species we just introduced. Washington State University just introduced it under permit from USDA, where it evolved with the apple. And it is thought that maybe this bee will be a little more productive in apple pollination. It's just being it was released by USDA, so it's just going to be released and tested this year. So it shows some promise. So understand that man's relationship with honeybees goes back a long time. This is about a 10,000-year-old cave drawing from France, northern Spain area, which depicts that overall relationship. But, you know, and that's what we did, hunter-gatherers for many years with bees, going in and stealing honey and brood for carbohydrate and protein sources. And it wasn't until about 1851 where we developed an understanding of an aspect known as bee space, which allowed for the removal of the frames and the development of the Langstroth hive, which is a type of honeybee hive we typically see. Now, the value of honeybees, I got this slide from the USDA, so it must be correct. They are extremely important in terms of their overall importance. Of special note is almonds. Almonds are by far the driver of this industry. And I'll refer to that and the implications that it has for you a little bit later. But the decisions that the beekeepers make of trying to get a crop pollinated in February in the middle of winter, and the demand for 8 to 12 frames of bees has some consequences. But also, you see a number of other crops that are equally important. So this is an important slide to be able to demonstrate. You know, bumblebees and mason bees and honeybees are like saying blueberries, raspberries, and loganberries. You know, they're different. And to say that all bees within, or all blueberries are the same, probably you would not agree with that if you're a blueberry grower. Well, the same holds true with honeybees. These are the species that I talked about. The larger picture is the Carniolan, which is, again, up in the Alps of Europe. The smaller picture on the top is the Italian. This is by far the most dominant species or subspecies of honeybee used by commercial beekeepers. Reason? Almonds. Italians just brood up. They just keep producing lots and lots and lots of bees to the point where they'll even starve themselves to death. They'll just keep laying eggs and laying eggs, so they're very, very strong. The carniolan and the Caucasian, which is the blacker bee at the bottom, are more temperature sensitive. They wait until there's a little bit of stimulus coming in before they start brooding up. But when they brood up very, very quickly and produce a good quality bee. The difference is, primary difference, is that the Caucasian and the carniolans fly at a much lower temperature than the Italians do. And you can see that anecdotally. There is some research being conducted by Washington State University this year to actually quantify that. But I have seen it time and time again where the Italians are not flying and the carniolans and the Caucasians are. So if you're just looking at brood count, oh, I got 12 frames of bees, this is great, but it's too cold and they're not flying, well, I'm glad you're happy. But you'd probably be much better off with colonies of carniolans and Caucasians at 6 to 8 frames than you would with Italians at 8 to 12 frames of bees. But you're not going to get many beekeepers doing that because they get paid in the almonds. And that's the primary difference. They want big colonies of bees in the middle of February when no bee in its right mind would be brooding up like that, except for an Italian. So there is an important aspect of the bee industry, which I'm pointing this out, is that we produce a lot of queens. If you notice down there, there are about 500 breeder queens. These are the actual queens that the beekeepers graft off to produce over 1 million queens a year. Some say it's a closer to 1.5 million, and they put them out in these little nook yards, and they go off on their mating flight. But one of the problems of that method, and these beekeepers are very good at producing large numbers of queens very quickly, is that we have a reduced genetic diversity. We have a very small genetic pool that we're grafting off of, and that's one of the reasons why WSU is bringing in stock under the USDA permit to increase the genetic diversity, semen only. So we bring in semen, we instrumentally inseminate them, we send them to USDA for testing for various diseases, and then once it's been released, then we can release them through the queen industry. But that has led us to a big problem in terms of trying to find answers other than chemical solutions to things like the varroa mite. So in terms of honeybees, queen, drone, worker, 3 castes that you're going to find in a colony. Queen is extremely important. She is the egg-laying machine, and she is the producer of a lot of pheromones. I found it chuckled a little bit when I heard about using spraying mandibular pheromone on your crop. It's meant to stimulate things inside the colony. So, you know, it just, and the queen is the producer of the mandibular pheromone as well as a number of other pheromones within the colony. She copulates in the open, in the air with up to 60 drones, typically around 15 drones is an average number of matings. But this is all meant for that genetic diversity of getting away from the colony, breeding with drones that have come from miles around and in a drone congregating area and getting that genetic diversity. She may copulate with 50, 40, 10, whatever number of drones, but the complement that she stores in her spermatheca represents only the equivalent of 1/2 of the semen found in one drone. But within her oviduct, it mixes, and there's a genetic diversity from all of the drones that she mates with. The point being, they breed in genetic diversity. That is part of the whole mechanism of how they evolve. And yet we have constantly reduced that genetic diversity in the terms of how we've done our selection and breeding. So I don't know if this is going to work or not. This is just this is one of the videos that this is going to work. Okay, but anyway, it showed the queen going off. It was pornographic bee porn of a bee going off on a mating flight in the air. It's kind of interesting. Now, a lot of people think that all drones do is eat, beg, and mate, and so we need to get rid of them. But again, it's stimulus. A healthy colony is going to have drones because they are trying to reproduce. It's the whole idea. We know that queens start laying when pollen is coming in. We know that the colony will have drones if there is pollen coming in. When there's no pollen coming in, they are ruthless. They kick these guys out unceremoniously kick them out as soon as nectar and pollen stop coming in. So a healthy colony is a colony with lots of drones. It's a mistake to think that you're going to remove drone brood and have more worker bees and therefore a better pollinating unit. You want a harmonious unit, and that includes having drones. And then there's the worker bee. The worker bee is by far the most important. They have various stages within their life. They start out initially cleaning the colony. They are nurse bees, the feeding the larva, the drone care, producing wax, nectar processing, because nectar will come in at about 20 plus %, and it's really not suitable for extraction or honey from a beekeeper's perspective or from the bee's perspective until it gets down below 15% humidity. So they process the honey. And then there's guarding the bees. You'll see a difference between guarding and bees that are aggressors. When you drive by on your tractor, there are one set of bees that will come after you. There's another set that are guarding. So they'll sit there with their front legs up, ready like this, ready to attack anybody who comes into your colony or tries to go into the colony. My wife was working some bees the other day where it was heavy yellow jacket predation. They were just coming in, and the bees were actually forming a bridge so that the foragers were coming in and being protected by these bridge of bees, keeping the yellow jackets at bay. It was pretty amazing to see. So this is the basic abdomen of the honeybee. The head has a number of factors. The ocelli, which you see on the top of on the forehead of the bee, that one, there's actually 3 ocelli, which help them identify the location of the sun and so with their orientation. The antennae, which they provide a lot of sensory activity, the hypopharyngeal glands, the mandibulars, and the proboscis, which is that tongue. Shorter proboscis on the honeybee is part of the problem. But they're ingenious in that they actually know, and not from your perspective, they know how to go around it and chew a hole in the side and suck the nectar out of the side, bypassing the anthers. That's one of the problems alfalfa growers and for some blueberry and other pollination. The thorax is really the driver machine of the bee. This is, you'll see the yellow jackets, they'll come in and they'll decapitate the bee, they'll tear off the abdomen, they fly away with just the thorax because it is a muscle machine. The legs are attached here, the wings are attached, extremely very important aspect. On the hind leg, you see the corbicula of the bee, or the pollen basket. This is a device used for storing the pollen. So once the bees pack the pollen on the corbicula, it's no longer available for the plant. They've spit on it, they've packed it down onto the corbicula, and they fly off. It's really the hairs on the bee, and you can see the feather type hairs in the lower frame there, which actually shows how this is where the attraction to the bee. Now, what's interesting is that bees actually develop a positive charge when they're flying through the air, static electricity. So then they land on a plant, the pollen will actually jump over onto those hairs to help stimulate it. And there's a number of other aspects which I'll mention during one of my videos that has no audio. And this is just showing the actual aspect of the bee. Wax producing, this is by bees that are getting on in their nurse age, maybe 2 to 3 weeks old. They produce copious amounts of wax secretion, and then the picture on below is the festooning of the bees. They'll actually, this stuff is just flaking off them in large amounts, and they'll pass it up as they're forming the pollen. If you go into a bee yard and you get out, you can smell the wax. That's a good sign. They are stimulated. Bees that are producing wax, yes, it costs honey, yes, it costs energy to do that, but bees that are producing wax are in good harmony, and that's a good solid colony that's working for you. So this is the bee space. I'm going to blow right through this. Pheromones, extremely important, but understand, I would say that 90, and some purists would argue that pheromones aren't a real sense of communication, but I would say that 95% of the bees communication is based on pheromones. The larva is younger larva produces a different form of pheromone than older larvae. So as the nurse bees are going along, they sense up this pheromone. They will detect whether they're going to feed a more dilute royal jelly or a stronger royal jelly based on the pheromone that they're actually getting from the larva. Throughout the life of the colony, pheromones are what dictates the bees to do things. But I don't think it's a good idea to try and just take the pheromone and think we can dump pheromone on something and it's going to be the magic bullet. There's specific pheromones that have specific tasks, and having the bees do things naturally, and the natural production of those pheromones will stimulate the bees to do what they need to do. The stinger. I always love the stinger. The stinger it's kind of shaped like this. So you have two lancets, lancets on either side that actually will work their way back and forth, back and forth, back and forth. And on the side of those, up inside the poison sac, there's actually little paddles. So as these lancets are working their way down with the barbs on them, it's just sucking the poison into you. Pretty cool. So I had to show this just because it just kind of makes people wiggle a little bit. But this is, a stinger going in, and you can actually see and the bees, once they sting, that's it. They're over with. That's a lot of their innards are sitting there now. So you can see the stinger actually pumping in the venom, and as little paddles are sucking that venom right into you as quickly as you possibly can. So the trick is, let me see if it actually comes up. Okay. This is a little, okay, I'm going to go in my wallet. I'm going to take out my credit card. I'm going to scrape off. Yeah, you get the poison sac off as quickly as you can. Just scrape it off, get it off as quickly as you can. And what I tell people, and people laugh, but when I get stung, I slap myself really hard, and then that hurts more than the stinger, and I forget about it. But the idea is get that stinger off as quickly as you possibly can. So the waggle dance, I don't know if the video is going to show, it's one of the more common types of communication we're used to. Bees are very good about determining exactly the source of where the pollen or the nectar is from their colony and providing that information back to the colony. They will get excited, stimulated, if there's a good pollen source, and they'll waggle more. Half, okay, half is 20. I'm in good shape. So they will actually communicate the information to the other bees in the colony. It's based on the nutritional and other needs within the colony as to whether they're going to one plant or another. It's just because this is a good honey source, maybe, but if there's a nutritional demand, more pollen is needed, there's going to be pollen foragers in there as well. It's a balanced approach in terms of what stimulates the bees to actually go after what they're going for. So anyway, unfortunately, this is a bee flying, and what I'm going to try and show here, and the narration doesn't happen. That is not a honeybee, but is the attractants that bees are stimulated by. This is going to show that bees are actually attracted by UV light, of actually being able to, this is the waggle dance, excuse me. I got ahead of myself. So the honeybee in this shot is giving their workers a taste of the nectar that they've just found, and then they do the waggle dance to actually indicate the location. So you can see that one is waggling, going back in the same direction. Here again, waggling. That very, very specifically tells the bees the exact distance from the hive to where the nectar source is. Very precise in terms of the communication that they provide to the other bees. The more she is getting excited, and the more bees that are in doing that, the more recruits they get to actually go after that particular colony. So this one is directly will be in direct relation to the sun. So fly right towards the sun, guys. Go 716 feet, and you have the nectar source that I'm telling you about. It's pretty cool communication. Carl Van French won a Nobel Prize, which typically biologists don't win Nobel Prizes for this discovery. So, and it's been disputed by Adrian Wenner, tried to say it just kind of gives them a general idea, but it's now been documented that honeybees are extraordinarily precise in terms of that communication. So And one of this is actually a shot of Apis dorsata, which is interesting that they, it's what videographers have to do. Now, what they're demonstrating here is that as the sun is moving about 4 degrees every hour, the bees have a built-in mechanism to compensate for that 4-degree movement of the sun. So they actually don't have to compensate for that. They automatically can figure out that there's a movement of the sun, and the waggle dance telling about it. And the narrator is far more interesting and concise in their presentation than I just was. But this portion of the presentation usually gives me the opportunity to take a break and a glass of water rather than trying to talk through it. But so So you have different foragers with honeybees. On each individual flight, you will have a pollen forager, you have a nectar forager, you have water foragers, you have propolis foragers. And propolis is a form of substance they get off of trees, which not only seals the colony up, but we now know that it has certain antibiotic properties. For instance, I now will go in with a grinder with a buffing wheel on it, a steel wheel on it, and buff up the insides of my boxes rather than having them nice smooth lumber. I'll rough them up so that the bees actually coat that with a coat of propolis, which has been shown to have certain antibiotic properties to it, and overall helps with the bee's health. But so once a bee gets stimulated on a given trip to become a pollen forager, they will stick with just pollen. They will not necessarily go out and get nectar and pollen, but there are those that go out and get both. But on that individual trip, they will be exclusive on a pollen forager and exclusive to that plant that they're on, be it dandelions or blueberries or whatever. They will stay with that plant. From the plant's perspective, that's a very good aspect in that you're not getting contamination of other types of pollen when you're actually doing the foraging. And this is what we're talking about. These are the hairs and the pollen grains. That's when you're getting your maximum pollination, not when the bee has put it on its legs. At that point, it's no longer available. It's that hair, the hair all over the pollen, and in the process, they're constantly grooming. When they're grooming, they're knocking off a certain amount of those grains that are falling on the stigma, or hopefully falling on the stigma, and facilitating the pollination process. Now, this is the one I started to talk about in terms of what bees see. And it's got a nice little ditty to it. I found a computer program that lets me steal videos off the internet, which is very cool. But what we know is that bees are very good about identifying floral sources. And they're just because we see things in the visual, they see things very differently. Back in the 70s, we were able to document that they see in the ultraviolet, and there's certain patterns on the plant, which you'll see in a second here, that actually helps them identify where the nectar and pollen sources is. So these are kind of landing patterns that the plant has put out saying, come in this way. And in that process, passing past the reproductive parts of the plant to help with the stimulated process. We have since learned there are a number of mechanisms that help in this whole overall process. We know that bees are more attracted to plants that produce caffeine. We also know that they're more attracted to plants that produce nicotine. We also know that plants develop a electromagnetic field that is decharged when the bee comes and visits. So if you'll see bees coming to a plant, if you go out and observe, they'll fly in front of this one, they won't hit that flower, they'll not hit that flower, and then they'll go to this flower. They are recognizing that, oh, by electromagnetic charge on the flower, that this plant or flower has been recently visited by another bee, and I have a better chance of getting a nectar or pollen reward by going to this flower. Really cool mechanisms that plants and bees have evolved over time. So I need to get into the topic is what's going on with the decline of bees. Well, there are a number of diseases. The varroa mite, which is in the top one, there's still a problem that a lot of beekeepers especially new beekeepers, don't recognize. And that's the middle shot. That is American foul brood in the scale form. It's very different in the scale form than when it's in the fresh dead form, but it can still be a major problem. And because of prophylactic use of antibiotics, many of the bacterial problems with honeybees are, we can't use some of the antibiotics like terramycin, which we used to use routinely. We cannot use any, well, we can, but it's not as effective as what it used to be. And then down at the bottom, we see results of deformed wing virus, and there are just recently there's a paper out within the last couple of weeks. There's now a third variant of this disease been found, which causes deformed wings, which is very closely associated with the varroa mite. So we've all heard about colony collapse disorder and what it is. Viruses, pesticides, there's all kinds of theories. For a while there, it was cell phone towers, it was all kinds of different things coming up as being the problem. Nutrition. Well, a study that was out in 2010, it's been out for a while, but it still pretty much holds true. There are over 61 variables associated with colony collapse disorder, none of which is the definitive cause. But what almost all bee researchers and most commercial beekeepers will tell you, the biggest problem we have is the varroa mite. The varroa mite evolved with Apis dorsata and Apis cerana, and then migrated over onto Apis mellifera, the honeybee. It is now, we used to call it, with the dorsata and cerana, it was Varroa jacobsoni. The varroa mite that's now on the honeybee as a new species, Varroa destructor, that does not go back to cerana and dorsata any longer. And loss of habitat. That is one of the second biggest problem that we have, is loss of habitat. You as growers want good, strong colonies in your fields when the bloom is happening. That just doesn't happen overnight. It takes a 3 weeks period of time for bees to develop. It takes a while for them to build up strength. And the beekeepers need places where they can go and put their bees where they bring in natural nectar and pollen and build up. So when they bring them into your yard or your field for pollination, they're already charged up, ready to go, and they're looking for a new crop, and you want them to land on your crop. Well, increasing habitat is one of the things we really have to focus our attention on. And then how we keep bees. This is one of the most innovative beekeepers that I know. He runs, his family runs approximately 140,000 colonies. Big time beekeeping. This is a picture taken about this time of the year in 2013. Maybe it was in January of 2013. Where I'm looking at, I didn't have my fancy iPhone that will stitch everything together, but I'm looking at 84,000 honeybee colonies all in the same location without a single flower out there. And they're there from September until February, when the almonds come into bloom. Costco gives WSU a lot of money for bee research because they sell a lot of bee pollinated products, and they're concerned about the decline of honeybees. Well, they insisted that they put, 84,000 hives. I want a bee suit. Well, they were the only ones that wore bee suits. The researchers that were there didn't have them on, and the bees started following the Costco people around with their white suits on because they're used to the sugar syrup trucks coming in and the guys getting out in their white suits and feeding large amounts of sugar syrup. So you had these Costco people with these balls of bees on their head. It was quite amazing. But industrial beekeeping, perhaps we need to rethink what we're doing. We know that when you feed only artificial stimulants, sugar syrup, artificial pollen, that there is a gene within the bees that helps them detoxify substances that does not upregulate. In other words, it does not turn on. So you are going from an area where they are being fed only sugar syrup into a condition in the almond industry where they're going to be exposed to toxins, and they have a diminished natural ability to detoxify the substances that they're getting in. So we need a lot more. Now, pesticide exposure. There are a lot of pesticides out there that the bees are exposed to, including miticides that beekeepers need to use in order to keep their colonies alive. If you do not treat your colony for varroa mite, it will be dead within a 2-year period. Anybody who tells you differently, well, I'm happy for you. But The USDA has found over 120 pesticides in colonies. It's something that we have to deal with and deal with a lot. Beekeepers, as one beekeeper told me, I can't make money off of dead bees. And so they have been forced to use miticides within the colony in order to control the varroa mite. And then there are exposures to agricultural pesticides as well as landscape pesticides. And one of the ones that's in the news most often, sorry about the font substitution, but the neonicotinoids are certainly out there and causing some concern. And this is a graph that actually shows the white dots are the number of bee colonies in the United States, and the black dots are the increased use of neonicotinoids. Well, from basic statistics class, you learn correlation is not necessarily causation. And just because there's an increase, I can understand why people think this, but it's not necessarily the case. Now, are neonicotinoids good for bees? No, they are not. They are an insecticides, and they do cause quite a bit of harm to bees. As levels as low as 0.2 parts per billion, very low exposure levels. So it is an important factor. So in order to test this, I actually went out and sampled pollen from over 130 colonies. We tested a total of 149 colonies throughout the state of Washington looking for neonicotinoids, where we actually went in, found the bee bread, which is the stored pollen within the colony, drilled a hole out, transferred it up to, put it in little baggies, put it in an ice chest, and transported it up to Tri-Cities, where they did analysis in terms of finding the compound within the colony. Well, we did find it in various areas, but we found almost none in rural areas, and that we used a 2-kilometer radius if you're within a urban area or in an agricultural area based on Washington State Department of Ag crop shape files. We listed you as rural. We found no significant difference effect within urban areas, but we did find within agricultural setting, both in bee brood, bee bread, and in beeswax, that there was a significant level of exposure to neonicotinoids, but we found nothing above the 3.9 part per billion levels. So what this really tells us, and the question we were asking, there is really no justification for banning the use of neonicotinoids, but there is plenty of concern that there are consequences for spraying. This is not something that is just innocuous and you don't have to worry about. So using and making smart application is extremely important. Today, for the first time, a new video that Lisa's in, I'm in, a number of other researchers are in, is being released. If you would like the copy of where the URL is, just email me and I'd be happy to send it to you. It's a 53-minute video that, other than having to watch myself talk for all that long, but it goes very well. Videographers are great. But we need to be concerned about neonicotinoids. We need to be concerned about all pesticides. We need to be concerned about fungicides, and understanding that, look at the ground. What's blooming on the ground? What is there standing water nearby? All those things. Bees need water. Bees need pollen. Bees need nectar. Be conscientious of where you're spraying, when you spray, and how you spray. And this is what I'm all about. I can do this because the bees are out of the colony and they have no defensive mechanism. So it's a very hammy thing to do, but it's my best way of being able to show you stimulate bees and they will respond in a predictable manner. So my get home message to you, don't necessarily cut your dandelions. Don't necessarily chop down all your maple trees. You're trying to stimulate the bees to go out and bring in nectar and pollen to get that queen to start laying, so that the bees have a larger demand for more pollen, so that they go out and pollinate more. So that's my presentation. Sorry about the high-winded aspect of it.
Thanks, Tim. We've got a couple minutes here for questions, and I'm going to start it off with a couple from Prosser again. The first one is, why is the press and much of the public so fixated on neonicotinoids being the problem causing colony collapse when the beekeepers know differently?
Your guess is as good as mine. I mean, it's simply a matter of, you know, to a large extent, a lot of organizations are using the honeybee and they dress up in little bee suits, and isn't it that to ban neonicotinoids? You know, no, I'm not a fan of neonicotinoids. They cause harm, but I grew up in a time when I was dealing with organophosphates, carbamates, chlorinated hydrocarbons, tells you how old I am, that where you have massive numbers of dead bees in your yard when you go in, massive numbers of dead bees. And so from my perspective, the neonicotinoids are a lot better than some of the older classes of chemicals. So.
Thanks. We've got another question from Prosser. Are there certain trees or plants in eastern Washington that are better for the accumulation of pollen for honeybees?
I would refer to Steve Shepard on that question. He's from eastern Washington. Did I dodge that bullet well enough? Sorry, I just don't know. So.
Thank you. Yeah, I wrote a paper about this when I was in school, covering a lot of the variables you cover, like nosema and IAPV and, neonicotinoids. And I know one of the characteristics of colony collapse disorder is supposed to be that the beehives are empty. They're found empty. Is that correct?
Well, yeah, that's part of one of the classic characteristics.
Okay, so obviously any biological organism has a lot of factors that influence the health of it. I'm wondering why specifically, especially the varroamide explanation as being the primary cause, why you'd see empty hives based upon that?
It's an interesting phenomena, but this phenomena of disappearing diseases or disappearing bees is not a new phenomena. Back in the 60s and the 70s, when I was working in commercial production, we used to call it absconding disease or a disappearing disease, where you'd come upon a colony and there wouldn't be any bees left in that. But I think it's a combination of management technique, the way bees are now managed, and in terms of the impact that the varroa mite has on the overall health of the colony. You'll start to see, if after 2 years, we call it snot brood. We don't, it just looks healthy. I mean, the whole colony starts to go downhill. As soon as an experienced beekeeper cracks the hive open, you can tell there's something not right here. And it's just a combination. So the mite is not only piercing and sucking the hemolymph out of both the larva and the adult, it's also introducing a lot of pathogens into the hive. So it's not an easy explanation that we really don't have an answer for. So, but why it happens and why bees don't come in and steal the honey, well, given the number of chemicals that a lot of these large commercial beekeepers use, that might be part of the explanation.
And I guess in reference to the Prosser question about why there's kind of a fixation about neonicotinoids, one of the things I was looking for and trying to figure it out myself, just an undergrad when I did it or whatever, was figuring out what variable kind of changed. And one of the variables is that the rise of the imidacloprid and the neonicotinoids in the mid-90s is correlated, and correlated is not causation. But with some of the decreases, and also in France, they banned neonicotinoids, imidacloprid, and they saw a recovery. So I think certain things like that, I mean, it's not as though there's nothing there, and you're saying that it's not as though there's nothing there, but, I think that, especially Penn State's been doing the research for like 10 years now. They're supposed to have come up with some kind of comprehensive report, and honestly, everybody wants a management solution.
I don't want to speak ill against a Big Ten university, even though I got my degree from the Ohio State University. But anyway, it is, there's a lot of work that's being done out there that is of questionable. And so, you don't believe everything you read, and you need to take the body of literature and take a look at a lot of different variables before you come to a conclusion. And so I don't want to disparage the researchers at Penn State, but there are some questionable things that have been published. And I think most of them will tell you it's primarily the varroa mite that's the biggest problem.
Okay, I think we'll move on to the next presentation.
Okay.
Thank you, Dr. Lawrence. So our next presenter